doing healthcare science. So we're studying human body, and you should know that our bodies are made of calcium and all these elements, and we have to study it to go further and deeper. If you want to understand the human body, you also need to understand the chemistry of it and how things react in your body, even if you went to study diet or anything like that. Okay, so obviously each periodic table, there will be different ones where you have the atomic mass number on top and the proton number on bottom. Sometimes is different, but just to give you a note that basically the bigger number is always the relative mass, atomic mass. The bigger number is always going to be relative atomic mass, regardless whether it's on top or bottom, because some periodic table, they just switch them. Okay, mass number is just proton number plus neutron. I think these are very basic, but please, if you have any questions, don't be shy. Okay, here's an example. This could be like a question where it's one of these multiple choice questions. So as you can see, it's saying, suppose we have 1 gram of hydrogen and supply of metals, lithium, sodium, and potassium. Assuming all react efficiently to form hydride. So these are hydrides. I don't know if you can see my mouse. Is my mouse appearing to you?
No. No,
you can't see your mouse. Okay. So LiH, as the product, these are called hydrides because it's a metal bonded to a hydrogen.
You can do annotation, I think, Rosa. That might be easier.
This is my first Zoom session, so I'm not sure.
Oh, please, don't worry then, don't worry.
So forgive me if I don't know where that is. I'll make sure I improve it next time.
Okay, no, it's fine. Thank you.
So here, for example, it's asking you should think which one gives you the greatest amount of hydride. So as you can see here, we have a line of an equation, and this is nothing to do with amounts. All we have is half a mole of hydrogen 2. So that means 1 hydrogen atom. Is everyone understanding this part? So we're not talking about masses yet, but the question is saying which one of these will give us the greatest amount of hydride. So forget what it says on grams. Can anyone think, how could you actually predict this?
See which gives you the most amount of product.
Yeah, so you have 3 equations, 3 different metals and a hydrogen in each of them. So which one you think gives you the greatest amount of hydride?
Does hydride mean just hydrogen as a product?
Yeah, the product, like I said, hydride means a hydrogen bonded to a metal.
Okay.
So basically a product. So which one gives you the highest amount of product out of these 3?
They're 40 grams. Potassium plus hydrogen.
Yeah. Why do you think it's 40 grams? Where do you think they got these numbers from?
Maybe that was the lowest weight as is to make it stable.
No.
Oh, no.
Think again. So think about the masses of each atom.
Oh, potassium's got quite a lot of mass.
Yeah, so if we go back to the periodic table, we go here at potassium, it's 39.
Oh yes, got the highest
plus
mass.
1 hydrogen atom is 1, so it gives you 40 grams as a relative mass.
Okay, I get that.
Yes, and for example, what did we have also? We had lithium.
And sodium.
Lithium is 6.9, so almost as 7. It's on the top left corner. Yeah. And then plus 1 hydrogen, it will give you an 8. So sometimes these things seem very simple, but if we got them in a multiple choice question, it will be mind-boggling. So here it's saying equal amounts of different substances mean equal number of particles, which we have here. All we have here is equal number of particles. We got one lithium and half of a H2, which means one H, and we got them there, right?
Yep.
So number of particle is still the same, but there are different masses. So atoms of different elements will give us different masses.
Yep, makes sense.
Mm-hmm. Then we go back to now to this is about the particles and the amount of atoms. So this is about Avogadro constant. Relative atomic mass of carbon-12 equals 12 grams exactly. So that's why they use it as a standard. If you ever looked at definitions, for example, of the relative atomic mass, it's always comparing it relatively to a carbon-12 that has the weight of a 12 exactly. If you ever looked at relative atomic mass definitions, you will see they always compare it to that.
Okay.
Yes, this will be recorded, and I'm planning to take it only for like 1 hour and a half or 2 hours, depending if more people join as well and people have questions, important ones. Like I said, you can open your mic and ask direct questions.
I'll probably only be able to stay for an hour, Rosa, just so you know.
Yeah, that's fine. Because I made these 2 hours slot free for me, so it's okay if anyone wants to ask anything. So relative atomic mass. So here, do you see this number 6.022 times 10 to the power 23?
Yep.
This number is called the Avogadro constant and is very important for questions when they ask you, what is the number of atoms or number of particles? So whenever you see a question that is saying, what is the number of atoms or particles? Always think about this number. It's always going to be about this number. This number, you multiply it by the mole. So for example, we have here half a mole of hydrogen 2. So if I asked what is the atom number of hydrogens existing here, all you do is just times that by this, the mole times this.
Okay.
Obviously, it would be more complex in some equations where you have to work out the mole from a different way. And then the last question would be like, oh, what is the number of particles? Okay.
Okay.
Okay. I think here's an example, how much stuff. Moles make understanding chemical reactions and interactions easier. Use relative atomic mass or molecular mass to calculate combinations. So that's exactly what happened in the first question, whereas asking, how did we get this number? It's just times in the moles by the grams. So the mole number is always the number on the left, not the small number. This is not a mole number, only that one. Anything on the left of a particle or an atom is going to be the mole number. Is that clear for everyone?
Yeah, that's fine.
Mm-hmm. Are you familiar with this equation?
Yeah, I am.
Okay.
Mass Mr. Moles.
Yes. It's just basically like to work out the moles. You have the gram that you have used, and divide it by the relative atomic mass that you can find in the periodic table. Obviously, these are just very informative things. It's about, for example, this person called Dalton, he was researching the shape and the structure of an atom, and they came out of different theories. There's a cheat sheet that you can find it on the page. and it shows you the history of how the shape of atom or the drawing of an atom have developed to what we have now. So units of atomic mass used in biochemistry is Dalton dA. Unified atomic mass unit is U. So Dalton is approximately equal to the mass of one hydrogen atom. So one hydrogen atom is 1, right? If we go back to periodic table, it's 1.0079. So basically just 1, kind of. So now in biochemistry, this is like a new thing even for me. We didn't study that in A level. So it's just saying that dA, maybe if you can write this down, dA, 1 Dalton equals mass of 1 hydrogen atom.
Okay. And how does that help?
I think it's when they try to use it in different ways. Like, for example, the mass of 1 hydrogen atom is just 1, right? So maybe I'm assuming that in some questions, they will say the mass of this is 3 Daltons. It's just a different name now.
Okay.
Does that make sense?
Yeah. Okay. Okay.
Maybe they will say 3.5 Dalton of NaHCl was used or something like that. And for people who didn't study very well or didn't look at their slides, they'll be like, oh, what's Dalton? I've never seen that before.
Okay.
It's like a new measurement unit, but it just means as it says here. So atomic structure. What determines the shape of a molecule? Why is it shaped this way? And what, for example, how you have a kink here, those kinks in cholesterol. Like, for example, you see it is going a bit straight and then it starts kinking. So it's all about double bonds. And also if you have aromatic rings or if you have electron pairs that are valence, or if you have space for more electrons to join. Also bond length and bond angles. And that's like, I think if I will make a revision session only to do with the shapes and bond angles and bond length, because this is from A level and it's a completely separate topic on itself. So if anyone wants me to go further into that, please let me know.
No, I think this is all right. This
Okay.
bit.
All right. So atoms are electrically neutral because number of proton equals number of electron. Is that fair for everyone?
Yep.
Yeah.
Yeah, okay. So neutrons, as we know, they are uncharged. They are in the nucleus. Protons, on the other hand, are always positive and also in the nucleus. Electrons are on the outside and they are always negative. So they are always on a series of orbitals.
Okay.
Okay. Why do elements combine the way they do? As I said before, 2Na, and then you have Cl2. So can anyone tell me which one is metal and non-metal?
Na is the metal.
Yeah. So basically, what type of bonding is this? It's kind of very obvious, but let's see.
Sodium disulfide.
Yes, but what's...
Or
No, it's
is
not.
it the sodium bridge one? Oh no, or like ionic or covalent.
Yeah, no, it's not covalent. Pay attention. So it's a metal with non-metal.
Oh, so it has to be ionic.
Yes. So it's ionic, where basically you don't share the electrons, but you are giving away. So you're losing and gaining. So one of them is losing electrons and the other is gaining electrons.
Okay.
So which one do you think is going to lose or gain? So if you thought about periodic table, right? You know how they form charges. So for example, here, you have group 1, group 2. What is the charge that group 1 will always form?
Negative. Also, yeah, it'd be positive because they need to lose an electron.
Yeah, so it's a +1, always. Group 1 in periodic table. So the group 1 is going down, just to make this more clearer. So groups go down, periods go horizontally.
Okay.
Okay, so group 1 will always form +1. Everything in group 1, H, Li, Na, K, rubidium, all of these will always form a +1 ion. Group 2 will always form +2 ions. It's actually good if you guys write this down because it makes things easier for you, just in case if you'd never studied it before. So group 1 is always +1, group 2 is always +2. Then you have group 3, which is boron, aluminium, and gallium. That group will always also have +3.
Okay.
Okay. And then we skip the carbon one, skip that group. And then go to nitrogen, phosphorus, arsenic. This will have a -3.
Okay.
And then oxygen, sulfur, selenium is a -2.
Oh, and then it just goes.
-1. And
Yeah.
then the last one is a 0 because it doesn't gain or lose. It's a noble gas group.
Okay.
Okay. So from that, we should always be able to know that, for example, let's see. So Cl, which group you think it was? Where was it on the periodic table? In the ones they form negative or positives?
It was on the. I'm just gonna get up the periodic table again.
Yeah.
It was on the left. No, it wasn't.
Chloride. Or
Oh, it was on the right. It was in.
Yeah.
So that means it's negative.
Yes. So what charge will it form, do you think?
-2
Yes. And Na, in which
is
group?
a positive one. So it needs 2 Na to counteract
Yes.
-2.
Yes, exactly. So that's why we have it the way it is, because they have to always complement each other. So if they are losing 2 electrons, the other one should be able to gain those 2 electrons.
Okay.
And so when you gain, you become negative. So who's gaining in this case?
The Cl. Chlorine.
Yes, Cl is the one that is gaining 2 electrons from Na. Na will lose them to form a positive charge.
And what do they just? How do they physically look? Just like together, like a Venn diagram.
I just need to learn where to write, actually. Should have looked at this before. Okay, one second. Here is whiteboard. Okay. Let's share a whiteboard. Okay. Can you see a whiteboard now?
Yeah.
Yeah.
So Na. One second, let me get my pen. It's not clear. Okay. So you have the periodic table on, right? So what does Na, how many electrons does it have?
It has 11.
11, okay. So 2 obviously in the middle. First, sorry. So this is a normal Na, okay? So what usually happens, why is it losing this electron? It's gonna lose this one because it's always trying to form a noble gas state, which means it becomes more stable because it doesn't have a valence electron at the outside.
Okay.
So once it loses this one, it becomes, this is the way you should draw this one usually. That's how they draw if you were ever to draw this one. So it become like this and with a +1. That's how they look together. This is what it's going to look like. That's how you draw this one. When you ask to draw ionic equations, ionic bonding, that's what it should look like. And then for calcium, how many it will have? How many does it have?
17.
17, okay. So, so you will have one second, where's the pen? Okay, you will have 2 obviously of this, yeah?
Yep.
And then when you draw the calcium, you just draw it here, like as it's on the side or somewhere wherever you have space. So this is, how many we have? 1, 2, 3, 4, 5, 6, 7. One second, guys, I'm just getting the charger. So we have, what did we have in there? 1 second, in presentation NA.
Na and Cl.
Okay. This one. So you see the red atom that I just put on there, it's coming from Na.
Okay.
So that the red electron, sorry. So because Na will lose, where is it? Na. So Na actually it's in group 1. right? So group 1 will form positive, like we said, +1. And Cl actually forms a +1, -1. So if you look at the periodic table, it's in the -1. Okay,
Okay.
does that make sense?
Yeah, that makes sense.
Yes, okay. And so to finish it, we just draw it like that, and then it's a -1. And we erase this. Okay, so that's what happens. So that one, electron, the electron here, this one is getting transferred to this.
Okay, yeah, I got it. Okay, thank you.
No worries. So this is how you draw it physically. If you were to be asked of ionic bonding, this is what it will look like, those two drawings. Okay, and now I should go back to clear this. Can you see the whole screen now or no?
No.
So how do we... Okay. How about now? Can you see the slide?
Yeah.
Okay. Okay, so what we did is basically looked at ionic bonding, but if you want to also have more examples, we can do more. Do you guys want to do, or do we want to move on?
I'm happy with moving on.
Okay.
Marina?
Yeah, same.
Okay. So recall any atoms of any elements are electrically neutral, as we said. So because, like, for example, now the ions that we formed, they should be charged with a +1 or a -1, right? But before
Okay.
they were neutral. Do we agree? So N2Na was a neutral one. Cl2 was a neutral. But
Okay.
together they started here. So when they gain or lose electrons, gaining an electron gives an ion a negative charge, which is called anion.
Okay.
And then losing an electron gives an ion a positive charge called cation. So which one was a cation in our case?
The cation was the Cl.
Pay attention. So losing an electron.
Doesn't it lose? Oh no, it gains it. Yeah. So the Na is the cation.
Yes, remember Na was on the left side of the periodic table, which means it's going to form a +1. So these are the positive ones. They always form a cation.
Mm-hmm.
Like this information that I'm telling you now should like sit solid with you to help you. It's like actually the basics of forming equation and knowing about ionic stuff.
Okay.
Okay. So if you just master the charges on the periodic table and start relating to the names, it will become easier. So, and which one was the anion, negatively charged?
Say the question again, sorry.
So which one got negatively charged and became an anion?
The Cl.
Yes. Okay. And here it's just asking, why do atoms lose or gain electrons? Why do you think that? Like, why do you think atoms will lose or gain? What are they trying to achieve?
The noble gas state.
Yeah, so they want to be more stable because these electrons will be like giving them a small charge will track them to somewhere else and they will keep moving and repelling. So electrons are organized into a series of shells. So you have shell 1, the blue one. Shell 2 is the red one. And where it says for you here, number of electrons, this is the capacity. So how much does each shell can take electrons? So basically, shell 1 will have the capacity of always taking 2.
Okay.
And second one is 8, third one is 18, fourth one is 32.
Okay.
Is that like obvious for you guys?
It makes sense.
Sorry?
Yeah, it makes sense to me.
Yeah. Okay, it's like all you have to imagine is like a cupboard. It's like saying this cupboard number 1 will only have 2 drawers. It can only fit 1 electron in each drawer. So like that basically. But then this is like GCSE knowledge and then A-level knowledge, but then also SPDF. Do you know what these are? Do you guys know what these are?
Subshells, aren't they?
Yes. So have you studied them before?
No.
No. Are they hard for you, or would you like
I
to
understood
go?
them when I think he said them when the lecturer was talking my
Okay.
arena.
Yeah, I did it before at A-level.
All right. Okay.
You know, when I move the screen, do you guys just see a presentation, or do you see the background as well?
No, just the background. Sorry, just the presentation.
Just presentation. Okay, so you're not seeing a Word document there.
No.
Okay, then I have to do that share it separately. Okay, so how do you change that? Okay, I want you guys to look at your periodic table, right? Like I'm looking here. The first two groups, these two groups, the one that starts with hydrogen and the second one that starts with beryllium, those two groups, they are called the S block.
Okay.
Okay, the S block. And then you have the block that starts from boron to helium. Boron, carbon, nitrogen, oxygen, basically those groups are called the P block.
Okay.
And the ones in the middle, scandium and to zinc, like all of them, the group from up to down, they are called the D block.
Okay.
If you were to search it on images, you will see how they are colored as well differently. So what happens with these usually is that they will always end when we try to put the electron configuration, when we write it, they will always end with an S if they are from group 1 and 2.
Okay.
Would you like me to show you examples or things like that?
No, it makes sense. It makes sense.
Makes sense, okay. And you know how to write the electron configuration if I gave you, for example, an atom right now? So
Go on.
So if I said, for example, can you guys write me the electron configuration for magnesium? As a practice, can you start doing that?
So, which elements are again?
Magnesium.
Okay. So you wouldn't.
The electron configuration, not the one with numbers, the one with SPDF.
So it'd be an S because you said the blocks are an S.
Yes, but how does it start? Do you know how they feel or no?
Probably no then.
Isn't it like 1s2, 2s2, 2p6, 3s2?
Yes, exactly, those ones. Because these could come as multiple choice questions, like a one marker, but it's kind of important sometimes to get the simple things. Oh, I found the annotation. Okay. Mhm. Okay, for example, so Hamza, are you looking? Look
Yeah.
at this one. So this is how they will always, one second. This is how they will always fill. You see this box that I'm gonna write here? This is how much electrons will be. So this one always will fill 2. 1s will always fill 2.
Hmm.
These are going to be in order, okay? If you can write this, it will be good for you later on, and we can even go further into it.
So 1p will always fill
6.
6.
2p, sorry, it's not this one. And they will always follow this order. So, in other words, s orbitals will only have the capacity for 2 electrons.
Okay.
And p orbitals, or subshells, basically orbitals, they will only have the capacity for 6 electrons.
Okay.
And d subshells will only have 10 electrons, the capacity for 10. Okay?
Okay, and they need to fill it.
Yes.
Otherwise,
Yeah, so for example, magnesium, it has 12 electrons. Do you agree?
yeah.
Yes, so you
So
agree.
it'd be 1s and 1d.
So it will be, no, like I said, they have to follow the order. Did you write them? Did you write them on a
Oh,
paper?
sp.
Did you write what I wrote earlier or no?
No, I missed that bit.
Okay, I'll write it again. I would like you to actually write this because it's following an order that will always be set. The only thing that will change is what's inside this box, and I'll show you 1 second. 2s. Okay, did you write this? Can you copy it?
Yeah, I am doing 1 sec. Okay.
Okay, and then you have 5f, 14. I think that's enough for now, but basically, okay, did you copy all of this?
Yeah.
Like I said, the only thing that will change is the amount of electrons. The electrons are in these boxes. And these boxes are called orbitals.
Okay.
Okay, so let's go back to magnesium. We said it has 12. Okay? Agreed?
Yeah.
Okay. So I'm gonna write how it should be written, electron configuration for magnesium, and then you will tell me if it makes sense. So it said 12, so pay attention to what's inside the boxes.
So
And
1 S2, 2 S2.
how many
2
now?
P6.
4, yeah?
Yeah, 2 P6, so it's 10. And then 3 S2.
Yes, so that's how many?
That's 12.
Yeah, so we stopped there. So now we have the 12 electrons of magnesium, and you see what it ended with, an S.
Yeah.
So basically, that's why I said this block is called the
Is
S
it S
block.
block?
So anytime you write the configuration, it will always end with an S, regardless whether it's 2S or 3S or a 4S, it will always end with an S.
Okay.
Okay. And remember, the shells will always follow the order that I asked you to write down. So what you do is fill the shell, the electron number in them boxes and stop. Don't write any extra. Does that make sense?
Yeah, that makes sense.
Mm-hmm. Okay. So now we talked about the S stuff. Got it out of the way. Obviously, we can, like, I can send you examples on them if you ever want to do more stuff. So now this all the way, it's just talking about shells and higher energies. Basically, will be a higher energy, the green one. But within these shells, there are subshells that we just called S, P, D, and F. Okay? And the
Okay.
orbitals are the boxes that I shown you. Like the boxes with electrons, how many electrons we'll have. Right?
Okay.
So if you can annotate it if you want. So the SPDF is called subshells, and the top boxes are called orbitals.
Okay. Got it.
So remember, electrons exist in clouds. Even though we call them shells or orbitals, electrons do not follow a defined orbit because they are rapidly moving, which basically forms the electron cloud. So if you know the new model, electron model, you know how complex it looks, right?
Yeah.
And it's all the electrons are moving on that track always in different directions. So that's what forms an electron cloud.
Okay.
So like, for example, this one here, the nucleus will be in the middle, and you see those bulbs. Basically, they are electron clouds. It's just showing you like how they form. It's like a region that has a lot of electrorepulsion.
Okay. Yeah. I've got it.
So here it's telling you each shell has S, P, D, and F subshells within it. Different types, different shapes and energies. So it's showing you here the first one, the red one, is the S shell. Is one orbital and 2 electrons in total. So because we have 2 electrons in S shell, it's kind of distributed into a circle.
Okay. Yeah, to keep equidistance.
Yeah, because they repel each other. And then when you have P orbital, it says 3 orbitals, 6 electrons. What it's talking about in orbitals, I would like you to draw this. I'm just gonna go into, come on.
Rosa as well. I'll only be able to say for like 2 more minutes.
Okay. So look, Hamza, these, remember how I said S has 2 electrons? This is how they look like inside the orbital. I would encourage you to write that down.
Okay. Yeah. Got that.
A P will have 3 orbitals. How many does a P have? How many electrons does it have?
2. Oh, sorry, 6.
Yes. Okay. Okay, so this is how they're gonna look like inside the orbitals. These are 6. Agreed?
Okay, yeah, equally repellent.
Okay, 3 orbitals, 6 electrons. Okay. And the D one will have 5 boxes, like that. And obviously, you know what I'm gonna do. It's just the same thing. The
Yeah,
electrons
and then
will.
just keep. Yeah.
Yeah. So that's how they're gonna look like.
Okay. All right. Thank you very much, Rosa. I've got to head out because I got a lecture soon.
Okay. Well, you take care.
Thank you. Thank you, Irina. See you in a bit.
I
Bye-bye.
see you later.
Are you staying as well? Do you want to stay?
Yeah, I'll stay.
Okay. So, are you familiar with the orbitals and how they look?
Yeah,
Yes, you've done A-level chemistry.
yeah, I did.
Okay, so it should be a bit quicker now. Okay, let's clear this. Clear all drawings. What is the hardest thing that you ever struggled with biochemistry?
I think like the stuff towards the end, because I used to have to miss the lectures because I used to have to go to work.
Oh,
So I'm like behind that stuff really.
okay. So are you familiar with these ionic bonding? How the attractions happen?
Yeah,
Okay.
yeah, I should be fine with those.
Covalent. Yes.
What did you say, sorry?
I said, how about covalent bonding?
Yeah, I think I'm okay with that as well.
Mhm. Let's see. Hydrogen bonds?
Yeah, they're okay.
Hydrophobic forces? Anything to do basically with water?
You
So
know,
go
I
over
think
that.
that was okay, but I had one question, you know, actually. It might sound like a dumb question,
No,
but
but
you know
go.
that first, you know that first question that we did at the beginning,
Yeah.
about that like amount of substance and stuff.
Yeah.
What was the answer to that?
Let's go back to it. This one.
Yeah, that one.
Okay, so it was for KH is going
Oh, okay.
to give you the greatest. But did you
Okay.
understand why?
Yeah, because it's got like the highest mass.
Yes, because potassium is 40 and one
Oh,
hydrogen is 1. So add them together is, potassium is 39 and one hydrogen is 1.
okay.
Yeah, so these questions, like I said, you know, the words they're just playing about, because if someone just looked at the question and forgot about mass, they'll be like,
Yeah.
"Okay, well, all of them are metals. We have half moles of everything. So what am I meant to do now?"
Yeah.
Yeah.
Yeah, that's fine then.
Mm. And you're good with these things, right?
Yeah.
Okay, so I'm gonna move on to the next slide, which is pH and buffers.
Yeah. Can we do that?
Yeah. Okay. So laws of thermodynamic. First law of thermodynamic, energy can neither be created or destroyed. Agreed?
Yeah.
Okay, so what happens usually is the energy. So what type of energies do we have? Do you know what type of energies we have?
Do you mean like I don't know how you mean.
Okay, so for example, you have heat energy, kinetic energy.
Oh yeah.
Yeah.
Yeah, electrical.
Electrical and waves and all of these basically.
Yeah.
But for now, we're focusing on heat energy in chemistry most of the time. And yeah, okay. So what we're talking about here is that heat energy, for example, I'm just putting it more specifically, heat energy can neither be created nor destroyed. So what happens is it's going to be transferred. So you either lose it or gain it. So in a flask, when you put things together, sometimes you'll feel a lot of heat straight away. There will be a lot of heat.
Yeah.
Yeah, so that heat is like being not created, but is being transferred from the energy of those elements that you put together. So what happens to it? It gets transferred to the surrounding. It gets lost. And some reactions you will feel coolness straight away. So here delta means change. We should always know that one. And E is for energy. So system plus surrounding equals 0. Why do you think that equals 0?
Because it's not like created or destroyed, so it's always transferred between the two states.
Yeah, so it can't be like a minus or a plus because it's not, it's just being moving around basically.
Yeah.
So then we move on to enthalpy. Enthalpy is heat transfer, and the reason why we have a delta H because it's a change of heat. That's what enthalpy is standing for. Heat transfer between a reaction and its surrounding at a constant pressure. Think of H as in heat as heat transfer, and the unit is kg mole minus 1. Do you want to note them down or?
Yeah, I'm writing it down now. Yeah, you can move on now.
Okay. So then we have enthalpy and bond types. So each bond will have a certain energy inside it. So you require something to break it. For example, if you had 10 sticks together, you require more energy to break them, right?
Yeah.
And these bonds will have similar energies. For example, you see the double bonds down here at the bottom, or the triple bonds, they will have higher energy required to break them.
Yeah.
Yeah. So energy is for the same bond will vary depending on molecules. So mean bond enthalpy is used. So for example, OH, it could be in water, but also it could be an alcohol, right?
Yeah.
So it will vary. That's why these numbers are not exact. So for example, there will be questions where it tells you, oh, OH, they will give you their own energy bonds. So these are not constant numbers. They will change sometimes.
Yeah. Yeah, I've got that all down.
Okay, 1 second. I want to move this one. Okay. So here is saying bond breaking is an endothermic process. What do you understand from the word endothermic?
Endothermic, like you need energy to do it, and then exothermic is like giving energy off.
Yes. So endothermic will take it from surrounding and it will become hot instantly inside the system, which the system we mean as in a flask or a tube or anything like that.
Yeah.
Okay, so delta H, a negative delta H favors reaction occurring. So delta H is just a change of heat. Laws of thermodynamics. All processes must result in an increase in the total entropy in the universe. Entropy is the word that means chaos, mess, separation, is how further things are apart, how further particles are apart. Does that make sense?
Yeah. So like a high entropy is like gases, isn't it, compared to like solids?
Yeah, so high entropy is the gas, like the highest one. Then you have lower is liquid in the middle, and the lowest one is the solid.
Yeah.
Okay, so there we go, entropy. There we have. So energy expenditure is required to maintain order and prevent entropy increase. So if you have something that is increasing into a gas, you have to maintain it. So it's like you have a mess and you ask the police to go and maintain order. So you require energy for that to prevent entropy increase. It's like entropy is a mess, or the increase or the change of from solid to liquid or liquid to gas. So if you want to go backwards, so for example, from gas to liquid, you have to put in energy.
Yeah.
Large molecules such as biomolecules have a very high level of order, so a very low entropy value. So remember, the word order means it's contained, it's maintained, it's stuck together, very close. And low entropy, that means basically it's like almost like a solid state. So biomolecules is like cholesterol or amino acids or tertiary structures that we have in DNA. And as you know, they are always, most of the time, they are in solid state. So they have very high order. Here is just basically relating things into the human body. It's just saying tissue organ structure are even more organized, so we have even lower level of entropy. Does that make sense?
Yeah, it does.
Because they are so compact. Entropy measures the randomness or disorder in a system. So we have delta S. So delta S is standing for like the randomness, how separated or how far away. That's what it's standing for. And it's measured in JK minus 1, mole minus 1.
Okay.
Okay. So here we have the Gibbs free energy change. I would like you to write this one down because it's like one of those really important equations, and sometimes you have to work out each one separately to find it out. Sometimes they will give you the delta G and you have to work out, for example, the temperature. And maybe work out delta S from something, delta H from something else. You remember when we said delta H is the change of heat?
Yeah.
For example, when you see a reaction and it says exothermic minus 4,000 something, it's this number here, delta H.
Okay.
Okay. So we have that. Entropy and enthalpy are therefore delta G. Also depends on temperature and pressure, like we said. It's convenient to know delta G is a reaction under standard condition. So these are the standard conditions that you should know. Well, I mean, it's not very different, to be honest. I think each system will have a different standard condition. For example, human body to room temperature is different because our standard temperature is 37, while the room temperature should be around 25. So it was important to see that in the question, what they're talking about.
Yeah.
And also here they tell you the pH, standard pH is also defined as 7. Let's call that delta G0 because it's standard.
Okay.
So here they gave us an equation which is about glucose plus oxygen. So what do you think this is, an endothermic or an exothermic? Obviously it's in there, but
Probably exothermic.
yeah, it's giving out heat basically.
Yeah.
Also, it's a good indicator when you see a condensation or a condensation reaction where it's like water being released. So it will release some heat with it. It's like sweating when you sweat.
Yeah.
Because you're releasing heat, so you release the water with. And so there
Yeah,
will
that
be
makes
like
sense.
examples like that, and I'm expecting them to give us some. I'm not sure, maybe in second year or third year, they give us more specific examples about human body.
Yeah.
So delta G lower than 0, that means negative. So remember this equation when we work it out and we get delta G. So it's telling us here is if it's lower than 0, if the number we get is lower than 0, it's negative, that means it's an exothermic reaction.
Yeah.
Okay.
Yeah, I've got it down.
And then it says if delta G is bigger than 0, that means it's an endothermic reaction.
Yeah, I've got that down as well.
If delta G equals 0, then the reaction is in equilibrium, which kind of makes sense because it's So it's also similar to the delta H, mainly impacted by delta H, because delta H, if it's an exothermic, think about it, delta H will be negative, right?
Yeah.
Which means lower than 0.
Yeah, so it's exothermic, yeah.
Yeah, that's why it's going, delta G will be affected by it and it will be lower than 0.
Okay.
Okay. So if it's equal to 0, that means it's in equilibrium. What do we mean by equilibrium? That means the forward reaction, it's the same, like we can go back to the backwards reaction. So they can continuously bounce back. So once A and B are forming, they form the product C and D, but C and D also bind to each other and go back to A and B.
Yeah.
It's almost like a chain reaction. Sometimes if something is in excess as well, it will keep forming something else. But in equilibrium, that means it will form the same thing that it was being produced. So go back and forth. So here square bracket mean concentration. This equation is also important.
Yeah.
I'm sure you went through this in A level.
Yeah, we did, yeah.
Yeah, and the units as well.
Oh yeah, you had to work out the units as well. Yeah.
You had to like cancel them out and stuff like
Cancel them
that.
out, yeah.
Do you remember these?
I remember that equation, but I can't remember the units now.
Basically, the unit of a concentration. Do you know what it is usually? Small dm3, right?
Yeah.
Okay, so for example, if you had to see each one of these with a mole dm minus 3, and you were to cancel them or something, that's what it's talking about. Like you have to,
Okay.
but some of them will have mole dm minus 6 or something. It depends on what they give you in the question.
Okay, so when will it be moldy and minus 6?
It's because okay, so you see those brackets together, they are multiplying each other, right?
Yeah.
So it's you are timesing the numbers by each other. So sometimes it will become a 6, and sometimes at the bottom, one of these A or B will have no concentration, so it will be just a 0. So it's almost like you're dividing. It's like you multiply the top and it became a -6, but at the bottom you have nothing, so the answer will be a moldy and minus.
Oh, okay, yeah, I get it.
Something like that, basically. Sometimes it will be one of them at the bottom and one of them on the top doesn't have a concentration or something.
Okay, that's fine.
Let me just write it down. I'm actually planning to put questions and put like a sheet, spread it around. So
Yeah, that'll be good.
revision on this equation and unit conversion. And maybe examples on Gibbs.
Yeah, that'll be good.
So here is saying delta G reaction equals 0, which means delta G 0 reaction plus RT ln K. So do you remember what does delta G means? Delta G 0 to the power of 0.
Wasn't that for like the
Standard.
pH?
Yeah, it was about standard conditions and pH equals 7. There is.
Yeah.
The standard free energy change for a reaction denoted as delta G 0.
Yeah.
So this basically equation is when something is in equilibrium. When our equation is in equilibrium, that means if you get a 0, that also means it's equal to that. It's like in math, when you give things, let's say you have an unknown and you say if x equals this, you know, and you have an equation and you say if x equals 0, then x also equals that. It's like if A equals B and B equals C, then C is equal to A. It's like a triangle.
Yeah, I get you.
Yeah, it's just saying that if the reaction, this part here, is saying if delta G equals 0, that means equilibrium. If it's equal to 0, that means it also equals this equation. So it's a new equation relating it to the equilibrium part.
Okay.
Sometimes these things we pass by them, but we just like, how do I use this?
I know, I don't think I even remember that equation, to be honest.
Which one?
This delta G reaction one, the one that equals 0.
Yeah, basically the one. Okay, so this one is only in equilibrium. They probably passed through it, and because you know how our lectures were, they sometimes were fast, and they would be like, oh, you know this from A level or something. But this specific part, I confirm it's actually new. I don't think it's from A level. I'm not sure. Don't remember.
Yeah, I didn't learn that at A level.
So you can write the denotions, what these mean, basically.
Yeah, I have done, yeah.
So what happens here, for example, they will give you an equation, right? And maybe they will give you temperature. Obviously, you will have R. Delta G reaction plus that. Okay, so they will give you maybe the delta G part and say work at temperature. Because K is a constant and R is a constant. So the only thing you can find out from this equation is either the T or this part, one of them two.
Yeah, you know, can I ask a question? You know, after RT, is that ln
Yeah, ln K, you know, in the calculator where you just put log.
Yeah, log, yeah. Okay, I thought I said in K and I was so confused.
No, it's ln. Make sure you put it as an L.
Okay, that's fine.
Okay, so rate at which equilibrium is achieved dependent on temperature, pressure for gas, reactant involved, and presence of catalyst. So these are just factors. And the position, if you remember when we used to have to write statements in A-level, the position of equilibrium will move from the left to the right. Do you remember
Yeah.
this? Yeah, it's talking about these specifically. Because remember, if the gas pressure increases or the pressure increase, it will favor the side with the less gases.
Yeah, that's right. Yeah. Because it
Yeah.
always like moves to like make it equal, doesn't it?
Yes, exactly.
Yeah.
And no presence of catalyst, but with equilibrium here.
Yeah, so the catalyst, that just makes it happen faster, doesn't it?
Yeah. It
Yeah.
makes it happen faster, but it doesn't affect the position at all.
Yeah.
Here is just showing you reactions inside the cell, because remember we're studying biochemistry, so it's biology, chemistry related to biology. So energetically favorable reactions, negative delta G, are used to derive unfavorable positive ones. So delta G, remember what it said, if it's negative, that means it is what?
Exothermic.
Yes. So a glucose, remember, it was an exothermic reaction.
Yeah.
Yeah, so we're doing it to derive a positive one.
Okay.
Wait. Exothermic reaction. Derive unfavorable positive ones. Okay, reaction. Okay, so remember here we said a glucose, this reaction is exothermic, right? But over there they said it is an endothermic. Why do you think that is? Basically, if you read here, it says is spontaneous. So spontaneously, without adding any energy, it will be an exothermic.
Oh, okay.
But here, these are saying energetically favorable reactions. So they are adding more energy or something to it to derive an endothermic one.
So these ones are going to add more energy to make it endothermic instead of exothermic.
Yeah, so see, this is glucose, but with phosphate, right?
Yeah.
One with oxygen.
Yeah.
It's very important to remember pKa is phosphate. Some people just forget what it is and think it's just a term.
Yeah.
Yeah, so this reaction here, it's different to that one. That one is glucose and oxygen to make carbon dioxide and water. But this one is making glucose 6-phosphate. And remember, these are, when you have phosphate, is about ATP and stuff like that.
Yeah.
Okay,
That
so
makes
you are creating energy here. So you are making heat. When you have energy, you are harvesting energy. Remember, it's also heat.
Okay.
So you're making an endothermic reaction will take from the surrounding, and it will be hot inside the system.
Okay.
Here it's an exothermic one because it's breaking the phosphate from ATP.
Yeah.
Yeah, so remember when you're breaking things, like important things, it's going to be exothermic, and this one is forming a new molecule. So yeah.
But when it says here overall reaction at the bottom,
Yeah,
would that be like endothermic or exothermic then, or would it just be like neutral?
I think this one is a neutral one. That's what I think. Because you add in an endothermic and an exothermic, so it's almost like you can go back and forth with this one.
Okay.
It's what happens with the body when you have glucose and ATP. If you studied the Krebs cycle in
Oh, yeah.
biology, because they go back and forth and they form each other again.
Yeah, okay.
Lots of small enzyme catalyze reaction steps, theoretically reversible and reversible. Okay, let's see, pH and buffers. So you know what an acid is, right?
Yeah.
So sometimes you will have a reaction and something is not an acid specifically, but it will act like an acid.
Yeah.
Yeah. So when something combined with water, it will release hydrogen. Maybe it's good if you write these down, even if you know them.
Okay, I'm gonna do it now. Have we got? Do we have a test on like everything from first year?
I have no clue, but I actually wanted to start this because even me myself, I got demotivated to
Yeah,
keep
same.
studying. So I thought, why not do it with everyone and encourage everybody to do so?
It actually was such a good idea.
Mm.
Because I remember before they said to us that it was supposed to have like an exam in like October, November time, but then I checked my timetable and I couldn't see one.
Yeah, it's just confusing. Even like me, like I think there were a lot of quizzes, but then at the end they were like, we're not taking, it's not gonna count seriously or something. I don't know if they said
Yeah,
they said that,
I
right?
did. Yeah, they said that it was formative, really.
Yeah.
I did them, but we'll see.
Okay, so here we have HA plus H2O giving us H3O plus A minus. So which one is a proton donor? So proton donor, it will give positive and it will make something else a positive, basically.
Is it the first equation?
Yeah. So like an easy way to suss this out is just see which one is becoming a negative. The one that becomes negative is always the one that is donating a positive. Because when you lose a positive, you become negative, isn't it?
Yeah.
Okay, so proton donor means it's donating a proton.
Mm,
In that case here, we can see that HA is donating the H, and the H is a positive one, isn't it?
yeah.
So A will become a negative here.
Yeah.
So that means the proton donor is the A. It's almost like a couple. It's almost like
Yeah.
a couple, and one of them is positive, the other is negative. And let's say this one, the A, is the one responsible to give away, to donate. So it donated the H to the H2O, hence why we have H3O. So it donated a positive to the other molecule. So A is the one that is acid, is a proton donor.
Okay.
Also, in this one, also an easy way to spot it, like I said, is see if it's negative at the end. If it's negative, that means it donated a proton.
Okay.
This one here, proton acceptor. So also in the first example, this is the proton acceptor, isn't it? Not this one, sorry. This one.
Yeah.
Because it has accepted the hydrogen from A.
Okay, yeah.
Mm-hmm.
So in the first one, H2O is a proton acceptor, and then A minus is the proton donator.
No, the A, not the A minus.
Oh, okay.
The A is the proton donor.
Okay. I kind of forgot this from A level. Like, I remember we did it for A level, but
Yeah,
looking back,
I know. It's been a while. It's very easy to confuse because when the question will ask you, it's very easy to put these down instead of the
yeah,
other ones. But you have to think about the original because we're asking the one who donated, so the originals in the original state.
yeah.
Yeah. It's similar here, so it's saying B is the base. Can you explain why?
Because it's gonna accept a proton from. Does it accept it from BH plus?
No, it's accepting it from H2O.
Oh, okay.
The dealing, always remember, the dealing is happening here in the first stage, like just here.
Oh,
So whatever they ask you, who's the acid, who's acting as a base, your answer should be from here, from the
okay.
originals. So B is acting as a proton acceptor because it accepted a proton from H2O.
Okay.
Because H2O is acting as an acid and it's donating a proton.
Okay.
It's just like you have to think about it like someone who's a charity and someone who's donating to a charity. So if I said, who is the base charity? And you say to me, B is the charity. And I say, why? And you say, because this guy right here, he donated a hydrogen to B. So if I said who's acting as an acid in the second equation, who would it be?
Would it be the H2O?
Yes, exactly. Because it's donating a proton.
Yeah.
This is hard for us because we're used about losing and gaining electron, but now it's different. It's about proton.
Yeah. Yeah, that's it. It's confusing.
Yeah, so just remember when it's acid and buffers, we're talking about protons and not electrons terms anymore.
Okay.
So here we have an example. So the conjugate base. Okay. So remember this one is acting as a base. Why do you think that is? It's a conjugate base, sorry. What does conjugate mean to you?
It's like, isn't it like a pair? So I think the base is gonna accept a proton from the same acid that donated it to them.
Exactly. That's why it's called a conjugate, because remember these are reversible.
Yeah.
So in previous example, right there, we only talked about going this way. We just talking about that. But now, all of a sudden, we have to also think, oh, so how do these right here donate or accept to these?
Yeah.
So obviously, why do we say this is an acid? First of all, let's establish this way.
It's an acid because it's gonna donate the it's gonna donate the proton to the CH3COO.
It's donating it to this one. Always remember the dealing is right here.
Oh, okay.
Okay. So once it donated, they gave us this product plus that one. Yeah,
Oh, okay.
these are just the product here. We only gonna talk this way now. So this one donated. How about this one? What is this one? Acting as what?
It's acting as a base because it's like accepting it.
Yes, so it's accepting, basically, accepting. Okay, so can you just delete this? Okay, now we're gonna go backwards now. That's like sometimes where it gets confusing. Okay, so we said this is acting as a base. Why is that? We said a base is, what's a base?
Proton acceptor.
Okay, so it's accepting from who? To go this way, to go to this state. Forget about the word acid here. So we have this ion here, and it's accepting a proton, but who is it taking it from?
Are you on about the CH3 C minus?
Yes, so this here is going
Yeah.
to accept a proton. Who is this one accepting a proton from? Who is giving the proton to it?
Is it not the CH3 COOH?
Yeah, CH3COO minus, this one, yeah? It's a conjugate base, which means it will accept an electron, a proton. So
Yeah.
it will accept a proton, but from who?
Oh no, we'll accept it from H3O plus.
Yes, this one.
Yeah.
So remember the dealing will always be here or
Yeah.
there. They deal with each other. And these, for example, this side will be a product or that side will be a product.
Okay, yeah.
Yeah, so this one then is called conjugate. What?
Conjugate base pair.
This one the base, this one the acid.
Or conjugate donor. I don't know the
Yeah.
word.
Yes, it will donate a proton to this one.
Yeah.
So here also an example. So when you say conjugate pair, it's not related to what we wrote here. Like it's just telling you that this became that, and that became that.
Okay.
It's like,
I
oh,
think,
this changed to this, and this has changed to this.
yeah. I think I'm starting to like understand it a bit more now.
Because it's very important to go like, oh, the first step is from there to there. Let's see who's acid and who's base on this side first.
Yeah.
And then decide the other way as well when they go that way. Who's acid and who's conjugate base and acid on that side. But then there's an extra layer, oh, conjugate pair. Sometimes people get confused and they like, oh, so what do I do here? Is it this and this because they're called conjugate? But it's actually just, oh, this changed from this or to this, basically.
Yeah.
Here, basically, we have that equation, similar one, where we said the products are on top. Remember that one? Products and reactants.
Yeah, I remember.
So this is it here. It's
Okay.
shown here. So we just did the products over reactants.
Yeah, I'm just gonna write it down. I don't get it though. You know, in the Ka, how come there's no H2O?
Basically, in Ka, whenever you have water, you just ignore it.
Oh, okay.
You pretend that it's equal to 1 or something like that. Basically, that's what if you have an old chemistry book from A level, you will see it will say just ignore water.
Oh yeah, I think I remember now. Yeah.
Yeah. This one also is an important equation as well, because you go back and forth from this one.
Yeah. Yeah, I think I remember doing that one as well for A level.
What did you say? Sorry.
I think I remember doing that last one for A level as well.
Yeah. Sometimes they will ask you to find out the Ka number, and they only give you the pKa number. So if you have the pKa number and you need to work out the Ka, you make Ka the subject, so it will become Ka equals 10 to the power of minus pKa. And you can find that in the notes that I'm going to provide, and they will be underneath the video in the description. So you obviously know about pH, right?
Yeah.
Okay, 1 second. Okay, it's just like telling you that pure water is around 7, which was the standard for Gibbs. Delta G0, pH
Yeah.
equals 7. Okay, here's pH is the concentration of H plus ions, only H plus ions. Remember that very well. It's about protons.
Okay.
So it's just about the concentration of hydrogen ions or protons, basically. Okay, so pH is the concentration of H plus ion, for example, as they are written here. Like, even though this is in a molecule, but remember this from our equation, where is it? Remember here, it's acting as an acid, right? Conjugate acid.
Yeah.
But it has been donated to, like someone donated a hydrogen to it, an extra hydrogen, that's why it's a plus. So it's counted as a hydrogen ion. Got it? So this is a hydrogen atom, a proton, and this one also, because this one has an extra hydrogen.
Yeah.
And it has a positive charge, so it's treated as a proton, as a hydrogen proton. So pH1 has a very high concentration of H+. Remember, acid in our stomach will have pH of 2, right? HCl, which is very, very acidic.
Yeah.
So the lower the pH, the more acidic it is.
Yeah, that makes sense.
Yeah. And here it's just telling you it's basic if it's the highest pH. That means it has low concentration of hydrogen plus. Maybe it's worth stating this one, right in this line. But maybe without this, but it's up to you.
Yeah, I've written it down now anyways.
Okay. Let me try to erase this writing here. It's not erasing it. Okay, just forget it. So pH is a logarithmic scale. If the pH increases by 1, the hydrogen decreases 10 times. So remember, when we say we go up, that means hydrogen concentration is going down, right?
Yeah.
So like we said here, if the pH number is very high, that means it has low concentration of H+. So the difference between each pH on the scale from here to here the hydrogen concentration is going by 10 times. So
Yeah.
-10, for example. Sorry, not -10, where is it? Yeah, 10 times.
10
So,
times.
yeah, 10 times. So define pH in the following formula. So here, this one, or this one as well, these are also equations to work out the pH. Because it's basically, if you want to write this sentence, let me just say, okay, just write on your notebook, working out
Yeah.
the pH
Yeah.
or the concentration
Yeah.
of H plus. So this is what you get from this equation. You can reverse it basically.
Okay.
So you either get the pH as your subject, and because you already have your concentration, you will work out the pH, or you can do it the opposite way. If you have your pH and you work out the hydrogen ions, you will do pH over minus log 10 equals H plus. Does that make sense?
Yeah, I think so.
So we're only reversing this way, basically. To get the concentration, we will do pH. For example, they give you a number of 7 or 14 or something, and you divide it by that. So pKa related to equilibrium constant dissociation of an acid. Half the acid molecules have dissociated, which means lost their H plus. pKa is the pH at which. Okay, so this is like a constant, pKa.
Yeah.
It tells you the conditions for it.
Yeah, I'm just gonna write those down as well.
So it's mostly
Thank you.
about, yeah, strong acids just consider usually as a form. Maybe you want to copy this if you want. It's up to you. Because there will be sometimes questions, and we got them in A level, but I'm not sure if we get them here. And there will be like, how define the strengths of acid based on the pKa number that you calculated.
Yeah, so is it a small pKa means a stronger acid?
Yeah,
Yeah.
it's similar to the pH matter. Like the smaller the number, the stronger the acid. The stronger
Yeah.
the pH. Yeah, the smaller the pH, the stronger the acid. But with this one, there are limits. So it's telling you if it's less than 2. Yeah, so from less than 2 is strong acid. From 2 to 7 is weak acid.
Yeah, I get you.
So there are more definition limitations with this one. It's more defined.
Yeah.
This equation allows calculation of pH from pKa and the concentration of acid and conjugate base. So it's almost most of these equations, as you know, you can manipulate them to make something the subject.
Yeah.
Yeah. So you can copy it down like it's, let me just see. Yeah. So this one is also like, it's like when you said the pH, you know, our first equation about pH equals minus log 10 equals H plus, right?
Yeah.
But this one also telling you, oh, there's another equation to work out pH as well. There's another way, and it's called Henderson-Hassel balch. So it's this one. You can copy this one down as well.
Yeah, put it down.
So you can just put another way to work out pH.
Okay.
There's so many equations in chemistry.
I know, yeah.
Like now, it feels like, okay, I understand this. But when it comes to a real question, you'll probably be lost.
Yeah, exactly, because you wouldn't know where to start from.
That's why, like, I didn't bombard this with questions. I wanted to go easy because if I put questions and everyone struggles.
Yeah, then there won't really be much point.
Yeah, at least now it's like, oh, okay, I see this, but I want to see how it works out in real questions. So then I can
Yeah.
research questions.
Yeah, that's good.
So what is a buffer? A solution that is able to resist pH change, useful for keeping biomolecules at the correct pH in the lab. And I'm sure we have that in our stomachs as well, because when it gets too acidic, you know, if you studied, what's it called, physiology, when we were studying the stomach, about certain cells when HCl is being released, if it gets too acidic, then something else will be released to buffer it.
Yeah, like an alkali.
Yeah, an alkali. So it will make the effect less acidic, the environment.
Yeah.
So it's a mix of weak acid and its conjugate base. A mix of weak acid, or a mix of weak base and its conjugate acid. I really want to remove this here. Why is it not going? It's maybe from this. Okay, there you go. Finally worked it out. Okay. So as we said before, let's see. This is like another example, basically. Come on. Okay, there we go. Okay, so what do you think of CH3COOH? What is it acting as?
It's gonna. I don't know because like it could be donating the proton to the CH3COO minus.
So it's, yes, it's a donor. So what is a donor?
An acid.
Okay, so this is acid. And this one obviously is going to be the base because it is accepting, see here? It has accepted the H plus.
Yeah.
Okay. So if this is an acid, that's a base. That's going this way, right? Now I'm gonna change the color and we're gonna talk about the red way, like going that way now. So CH3COO minus, to go from here to there, to this state, is it accepting a H or is it donating a H? That way?
It's donating one.
No, from this state to that one.
Wait, from CH3 to H2O.
No. Look at, do you see the red marker?
Yeah.
Okay, let me just erase everything. Okay, from this here, this way, to that
Yeah.
one. Is it gaining a H or losing one?
It's losing one.
No, pay attention. So we're going from here to here, from A to B. Look at it, CH3COO minus, and it's becoming a CH3COOH. Is it gaining a H or losing a H?
I don't see how it's gaining one.
Look, it's a CO minus here. And there it's gained one.
Yeah, so it's lost one from there, from A to B. Because
No.
that B one's gained it, hasn't it?
No. Let me just open a new page, maybe. Okay. So our equation originally was CH3 COOH plus H2O. And going this way, we have H3O plus and yeah.
Yeah.
Okay, this way we said this is losing, so it's donating, so it is an acid, right?
Mm.
This one we said it's a base.
Yeah.
Now we want to reverse it. So we are reversing the roles now. We're going from here to there. Okay? So I'm
Okay.
gonna write it here, but from the other side. So we pretend those are our originals. Okay.
Okay.
So let's pick a color green. Okay. So who's acting from this side? Okay. CH3COO minus, and it became CH3COOH. Is it gaining a proton or donating one?
Oh, wait, I think I get it now. Is it gaining one?
Yes, so it's accepting. So if it's an accepting, that means it is acting as,
A base.
yes. So here we call it conjugate. I'm just gonna put con and base, yeah?
Okay.
Because we're going that way. We're asking about that way.
Okay.
And this one, obviously con acid. Yeah,
Yeah.
makes sense because here, look at it, it's a plus and it lost a H. So
Yeah.
it has donated. And acid is a donor.
Proton donor, yeah.
Yeah. So I think what confused you is because I was talking in the same line.
Yeah.
Yeah. But this is how it's going to be in the exam. That's why, unfortunately, it's a bit.
Yeah.
Yeah, it's just like you have to forget what you wrote first there and pretend you're going from here to here.
Yeah.
Yeah. Okay. So what are we asking here? Mix of weak acid and base. Okay. So which are conjugate pairs?
Um.
In this line. So this one, what is the conjugate pair for it?
Oh, I don't know.
Okay, it's about itself, basically. It's how it transferred, remember that. And this one, it's about itself.
Oh, yeah.
Yeah,
Okay, yeah, I get that now.
I think we got it somewhere here. Remember this?
Oh, yeah.
It's forget about these things. Basically, when you write this one, it's about which is the same as it is, like how did it change, basically.
Yeah, that's fine.
Yeah. Sometimes it's confusing. Like I said, even at the start, I said when people see the word conjugate pair, they will think about what they wrote from the second side of equation,
Yeah.
and they get confused, and that literally happened to you 10 minutes later.
I know, yeah.
Okay. Where are we now? Our concentration of H plus in solution affects processes, molecules, and reactions in both in vivo and in vitro. So in vitro is like the lab one, and in vivo is like in the body. Yeah.
Yeah.
So if the cell inside your body while you're still alive, it's called in vivo. And in vitro is when they transfer a living cell from your body and take it into a lab.
Okay.
Effects from in which protein, including enzyme, exist on other molecules. Type control of pH needed in the laboratory and physiological buffer systems. pH in human body is maintained between 7.35, these numbers are important, you should always know them, and 7.45. Outside this range, you will have coma or death. That's what happened with the blood as well. The buffer is the CO2. Now we have buffers in laboratory. Okay, so we have HA, that's our molecule. Remember we said the acid, yeah, it's acting as an acid. Remember we said A is acting as an acid because it has donated the H.
Yeah.
Okay, so we have our acid. And this one is the conjugate pair of that one, isn't it?
Because it's accepting it.
No, remember what conjugate pair mean? Conjugate pair.
Yeah.
It's basically the molecule and what happened to it afterwards.
Okay.
Because we're talking about HA. I don't know where's that. Yeah, that example here. So the conjugate pair for HA is the A. Yeah?
Yeah.
Remember, it's the molecule and what happened to it afterwards. It's a pair.
Okay.
Because I didn't say conjugate acid or conjugate base, I said conjugate pair. So it's like very, like need to just pay attention to that one. It's because sometimes. So we have the conjugate pair here, those two.
Okay.
So that's the acid one. Okay. And buffering zone. So here, basically these graphs, I had a document that we took from a revision session. I don't know if you ever attended that one while we were at uni.
I can't remember.
Okay.
Was it a lecture?
No. It was a revision session that you can book yourself or something.
Oh, I didn't even know about that.
Okay. 1 second, sorry.
It's
just
okay.
over 2 o'clock, that's why.
Oh, you can go if you want.
I think we are finishing anyways. These ones are just examples and summary.
Yeah.
That one.
Oh, we got through it all then as well.
Mm.
That's good.
Okay. Well, I'll be sharing more documents of like questions, and I'll share.
Yeah.
Hello.
Hi.
Yeah. I said I will share them on the group, possibly in the 2 coming days, hopefully.
That's fine.
I hope you had a good time.
Yeah, you've taught me a lot. Thank you.
No worries.
Okay, take care.
You too. Bye-bye.
Bye.
Bye.